we're going to be covering transcription and translation. By the end of today's lesson, you should be able to define RNA, contrast the structure of DNA with RNA, contrast the function of DNA with RNA, and define both the processes of transcription and translation. Now, we already said that information moves in one direction, basically from DNA to RNA and then from proteins. Essentially, the information is all found in DNA. We know that DNA is like the blueprint. It has all the information that pertains to the organism and all of its processes and structures, etc. But in order for this DNA to be used in order to make a protein, which we know proteins are extremely important, they determine essentially everything that is going on in the organism, whether it be all the processes, they act as enzymes, they act as catalysts, they determine traits, right? So they basically are used and found in virtually every part of your body. In order to take the information that tells us how can we make this protein, in order to take this information in DNA and actually make the protein, we first have to code the information as a kind of intermediate form, or as like a middleman form, which is known as RNA. In other words, the information cannot be used directly in DNA to make protein. It is not in the correct language. Let's put it that way. First, the information of how to make this protein has to be translated in a language that's going to be understood by the structures that are going to help us make the protein later on. And this form is known as RNA. It's very similar to DNA, and we'll talk about that, the similarities and differences next. So making protein is also known as protein synthesis. To synthesize is to make something, and it happens in two steps. Transcription, which, like I said, basically takes DNA and changes it into a different form known as RNA. And then translation, which reads this new form of information known as RNA. It reads it and it makes sense of it, it interprets it. And then it basically uses amino acids, which we know are the building blocks. They are the monomers of making protein, and it uses those building blocks to actually make protein. Okay? If you remember in the past, we said that a monomer of protein, right, the smallest building block of protein, is an amino acid. And that this monomer, like a bunch of these monomers, come together to make a protein. The protein is the polymer. So this is what we mean by the RNA basically contains information in terms of it tells the body which amino acids, which monomers to pick up, to use in order to make the protein that it needs. Okay, so making protein synthesis is slightly different depending on if the organism is a eukaryote or prokaryote. In other words, if the cell is prokaryotic or eukaryotic. Now, in the eukaryotic cells, we know that eukaryotic cells or eukaryotic organisms are a lot more complex. Eukaryotic cells have a lot more organelles. They do have a nucleus. So therefore, the processes that basically are found in protein synthesis happen in different locations. Okay, so replication, copying DNA, which we talked about yesterday, and transcription both happen in the nucleus. And then translation, which is the last step of protein synthesis, the last step of making proteins, is found or happens in the cytoplasm. Okay. On the other hand, because prokaryotic cells are so much simpler and they don't really have a nucleus, everything is going to happen in the same location. So replication, copying DNA, and both steps of protein synthesis, transcription and translation, all happen in the same location at the same time. So they're all happening in the cytoplasm at the same time. Now let's talk a little bit about RNA. We remember that DNA stands for deoxyribonucleic acids. That's where the DNA comes from. RNA is a ribonucleic acid. So essentially what this means, it contains a different sugar, okay, a different molecule that makes it up than DNA. It's just like DNA. It is made from nucleotide monomers. So if you remember, both RNA and DNA are nucleic acids, and therefore they are made from nucleotide monomers. And like we said, its function is to act as a link between the DNA and the nucleus, and finally the protein that is made in the cytoplasm. So if you remember, in eukaryotic organisms, in eukaryotic cells, transcription happens in one place, in the nucleus, and making the protein, translation, is happening in the cytoplasm. So the DNA and the RNA are both found in the nucleus, while the protein is made and found in the cytoplasm. This is what I mean here by the last point. Okay, let's compare and contrast both of these structures. So we already know that they're both made from nucleotides, so that is a similarity. They're both found in the nucleus. That's another similarity. But in terms of differences, like I said, they structurally, they look very different. So RNA has one strand, as you can see from the picture, while DNA has two strands. RNA is made from a ribose sugar, a specific kind of sugar, while DNA is made from a deoxyribose sugar. It's a different kind of sugar. Another thing that they differ in is the bases. So if you remember, every one of these lines is a base, and they kind of act as the steps of this double helix or ladder. And in DNA, the bases are A, T, C, G, okay? However, which stands for adenine, thymine, cytosine, and guanine. So essentially, every one of these is either going to be an adenine, or a thymine, or a cytosine, or a guanine. The order really depends on the organism, and that's where all of the genetic diversity that we see comes from. On the other hand, in RNA, like RNA will still have the adenine base. It still has the cytosine and guanines, very similarly to DNA, but it does not have thymine. Instead of thymine, RNA has a different base known as uracil. So I'll write it here so you see how it looks like. Okay, so that U that you see stands for a different base known as uracil. And the last structural difference that we talked about is that DNA is a double strand, while RNA is a single-stranded. Okay. Now, there are three main different types of RNA, and we're not going to go into details about what each one is, but what I want you to know is that each one of these types of RNA is going to be used to perform a specific function during the process of protein synthesis. Okay, each one does a different role or a different job, and if you remember, we always say that structure is related to function. So since these RNAs have different jobs, then you can note that they definitely look very different. They have different structures, because remember, structure determines function. The first one is messenger RNA, and the main job of messenger RNA is to carry the information that is found in DNA to basically in the nucleus to specific organelles in the cytoplasm, which are the ribosomes. And I want you to kind of try to remember, what do ribosomes do? What was their job when we learned about the different organelles found in the cell? If you remember, ribosomes, their main job is to make protein, right? So here we have it. Where do they get the instructions for making protein? How do they know the recipe? Okay, they need, just like we said, ribosomes could be, we could think of them as like chefs, right? They make proteins. Where do they get the recipe from? Where do they get the instructions from? Well, here you go. Messenger RNA carries the instructions, carries the recipe from the DNA that is found in the nucleus outside to the cytoplasm so that the ribosomes can use it to eventually make protein. The second kind of RNA is ribosomal RNA. And just like its name suggests, ribosomal RNA, where do you think it's going to be found? Definitely in the ribosome. Okay, so it basically it makes ribosomes, because you need that you need that ribosome to make the protein. And that ribosome needs to be made by something else, which is ribosomal RNA and protein. So do you see how like everything is related here? Without protein, the ribosome wouldn't be able to be made. And without the ribosome, the protein wouldn't be able to be made. So it's kind of like a chicken and egg situation, right? They need each other to happen. Finally, transfer RNA, tRNA, its job is to bring the amino acids, the building blocks that are going to be used by the ribosomes to make the protein. So remember, the chef, the ribosomes, cannot make their final dish, cannot make the protein without the ingredients. And in this case, the ingredients are the amino acids, okay? So the ribosome reads the recipe that was brought by the messenger RNA, okay? And once it reads the recipe and figures out which amino acids it needs, the transfer RNA is going to bring those amino acids so that the ribosome can use these amino acid monomers to build an entire protein molecule. So transcription is the first step of this entire process that I'm talking about, known as protein synthesis. And its main purpose is to produce a complementary strand of RNA by copying a complementary strand of DNA. So in other words, if I wanted to simplify what that just said, transcription takes a DNA strand and it makes an RNA strand using it. Obviously, it needs some help. Every little process in the body needs help. And the enzyme that helps in the process of transcription is RNA polymerase, very similar to DNA polymerase, which we talked about yesterday. RNA polymerase basically binds or adds the nucleotides together in order to make that RNA strand. And the location of transcription, like we mentioned before, is in the nucleus. All right, so the next step is translation. Once that RNA strand is made, it's going to leave the nucleus, head to the cytoplasm, where the messenger RNA is going to take that message that was in the RNA strand, leave the nucleus, and head to the cytoplasm, where the ribosome is going to read that recipe, read that message, and translate it into a protein. Okay. If we wanted to kind of summarize this whole process of protein synthesis as a big picture, this image does a very good job of summarizing everything. So this whole process is showing protein synthesis, making protein. The first step of protein synthesis is transcription. What happens in transcription? Transcription is when you have a DNA strand, okay, a DNA strand, basically written, transcribed in a different language, a language that the body can understand, which is RNA. So it basically, you're going to take a DNA strand and you're going to make an RNA strand during transcription. And obviously with the help of specific enzymes, etc. And you'll notice that in the RNA strand, we don't have thymine anymore, okay? That has been replaced with uracil, a different base, a different nucleotide that is found only in RNA. Once that strand of RNA is made, it can't really leave the nucleus. Instead, a messenger RNA is going to basically carry the message that is written in this RNA strand, messenger RNA, and is going to leave. It will go to the cytoplasm, and the ribosomes are going to read this message. They're going to read the message on the RNA, on the messenger RNA, if we had to be more specific. And the thing is, when you're reading something, when you're reading, like when you first started learning how to read in KG or grade 1, or whenever you started to learn how to read, you would read it letter by letter, right? That's how you would learn how to read. You would read letter by letter. Well, messenger RNA doesn't really read letter by letter. Instead, it reads in threes. So it basically will read this message in threes, okay? So AUG would be one message, AAG would be another message. And each one of these three combinations of nucleotides is known as a codon. Okay, this is what we mean by that word codon. And every codon is a recipe for bringing one amino acid. Okay, so every one of these is an amino acid. So essentially, in order to know which amino acid we need to bring for this recipe to work, the message is going to be read in 3 in codons, and every 3 will tell the body to bring a specific amino acid. At the end, all of these amino acids that are brought are going to be combined into a huge chain, which is a polypeptide chain. In other words, a polymer of amino acids. In other words, a protein. Okay? So we go from DNA to RNA in transcription, RNA to a protein in translation. And that is about it. That's basically protein synthesis in a nutshell. I hope that was clear. And as usual, if you have any questions, please let me know.